Hi, sorry, I'm just setting up right now. Just getting it all going. Hi, guys. Welcome. I'm gonna try my best to stay with you tonight, but I won't stay too long. As you can hear, my voice is totally going. I think I've been to too many sessions. But I want to make sure that I do obviously help you guys as much as I can. Hi, guys. Feels like you're just sitting there waiting for me to jump on. Um, do I need to know about histone modification? I had to think what you were saying there. Histone modification, you have to know about transcription factors. If they're going to give you any question about that, they may actually, in fact, let me bring something up earlier. They may give you the information. The reason I'll start this session on this. I was going through with one of my private clients tonight, some questions, and we just happened to be looking at the question on, no, it's fine. Honestly, don't worry about it. We were looking through some questions to do with enzymes and DNA, etc. So one of the things that we were looking at, which was in the 2023 AS paper, was looking at the fact of they put this information out there. And when I was doing this with my client, I was like, so they're not in year 13, they're in year 12 right now. So they've got another year to learn all this stuff. I was like, I can so see where this could go for an A-level question rather than the AS paper. So I'm always telling you guys to make sure that you do practice your AS papers alongside the A2. I know we've not got enough time, but what I would say to you is by doing that, you can see where some overlap so they can take some of the questions a bit further. How can make the most of the remaining time? Focus only on the stuff you hate, only on stuff that you really would dread. If you would cry if that topic area would come up in your exam on Friday, study it. Do that stuff. Don't worry about this other stuff. Don't worry about the stuff that you know. Don't worry about the content where in your head it's already there, it's ingrained. It's the stuff you don't know that's going to throw you off. And the more you practice the papers, the more you practice the questions, the more likely you start to see where they could go with the question. So I never just answer these questions. What I want to do is look at where could they go with a question, because you're never going to get that same question back again. What you're going to get is different versions of the question. Yeah, module 6 is hard. Paper 2 is always harder for most students. So it's really important that you note that paper 1 is done. Paper 2 will be either really hard just because the topic areas of module 6 is in there, or it might be a nice paper and paper 3 will be hard. There's always two papers that are harder than the other one. Just depends where they're going. Paper 1 wasn't the easy one, so let's hope paper 2 or paper 3 is. Right. The reason I want to talk about this is because you mentioned about histone modifications. And this question, they actually spoke about it. So here they're told that there's enzymes, and these enzymes are going to catalyze the removal of CH3. So they're catalyzing the removal. Tony, I will answer that question in a second. Let me just finish my train of thought on this one. So they catalyze the removal of CH3 groups from cysteine bases in DNA. So in this question, they've given you lots of information. What they're doing is they're seeing, can you pull out of this information to then apply it? So what we're talking about here is that the rate of transcription is going to increase when this group has been removed. So this question was obviously testing AS content. How could they bring it into A2? A2, what they could be looking at is, do you understand that this is acting as a transcription factor? In the fact that this enzyme is going to catalyze the removal of the methyl group, and therefore, what are we talking about here? We're talking about transcription factor. Now, for those of you who are just joining me, I'm not answering this question, just to be clear. I want to show you where they could go and what it could actually do with regards to the AS content in module 6. Right, Tony, let me see what you said. Do we need to know the role of cyclic AMP in the lac operon? So you need to know the role of cyclic AMP and the fact that cyclic AMP is going to activate other enzymes, protein kinases. You don't need to know any more than that. So they're expecting you to understand that cyclic AMP is where there's going to be a transfer of a phosphate. So that phosphate is going to activate this protein kinase. And this is where we're doing post-translational modifications. This is where the gene expression comes in. Right. Amna, what have you asked for? Question 19, paper 2, 2023. Right, let me have a quick look at that one. And then question 2. Just to check. But on why are you guys seeing that one? It's not the one I want you to see. You should be seeing. Why is it showing like that? That's so weird. Okay, hold on a second. Sorry, guys, on TikTok, give me a second. For some reason, it's showing you the wrong one. Just add this in. Here we go. That's the one I want you to read. Here we go. Right, what did you say? Sorry, Amna, you said question 19B. Let me have a quick look. Okay, this is about the clonal expansion, clonal selection part. Here. This one. Why is it doing that? TikTok is really not liking me tonight. What's going on? Sorry, guys, give me 1 second. I'm going to end the live and I will restart the live in a second. Oh. Right, okay. Think what's this? No, I've done it again. Right, I can't do that on TikTok, so I'm going to have to just say goodbye to TikTok. Sorry, guys. If you're on TikTok, I do apologize, but here we go. Okay, it's not working, so we're going to just roll with it on here. So I feel bad for those on TikTok, but I cannot make it work on there. So you're just going to have to listen to me as I'm through this. Right, okay. So sorry, I'm not back to this one. Okay, so you had this question, and I'm guessing you were struggling because it wasn't working, on how to actually figure out the clonal expansion and clonal selection part of the question. Is that where you were struggling with? So methylation, phosphorylation, no. When we're talking about the methylation and we've got the transcription factor, you'll be talking about that as being the transcriptional level. So we're talking about transcription factors, we're talking about the transcriptional level. If we're talking about the actually what you have said, sorry if you're just joining me again. And if you are talking about the changes to the histone proteins, the proteins being modified, then that would be post-translational. So it's kind of like it could be transcriptional or it could be post-translational, depending on whether it's asking you about the protein being modified or whether it's asking you about something that's activating the transcription process. So it kind of links to two. Right, Amna, on your question, I don't get why B and D is high T helper cell activity. B and D. So what's happening at this point here then, looking at this question, what you've got to realize is you're looking at your antibody production changes. Just here. So because we've got our antibody concentration is increasing, we know that we have more cells. And because we have more cells, that therefore is going to mean that there's going to be more protein synthesis occurring. More protein synthesis results in an increase in the antibody production. So we know we've got clonal selection that's occurred during A after the exposure. We know that we've then got expansion to get more cells. And then we need to look at the fact that it's asking us how high T helper cells. Well, if we've got a lot of antibodies being produced, those antibodies are coming from B cells. Those B cells had to be activated by T helper cells. So the T helper cells encounter the antigen presenting cells. What they'll then do is they will then activate the B cells by releasing their interleukins. So it's important to note for clonal expansion, your T helper cells are going to be releasing those interleukins, those chemical messages, so that they are activating that increase that's going on there. So in that case, the reason it's looking for B and D is because without the helper cells, you wouldn't be getting the B cells to go through expansion and then differentiation into the plasma cells. So, Tony, to answer your question about clonal selection, clonal expansion, clonal selection is selecting the correct lymphocyte with the right antibody, which binds to the antigen. Clonal expansion is where you're going to make more of the cells. And why does E have the highest number of memory cells? You put C. Interesting. Well, if we've gone through clonal expansion each time, so you should have put clonal expansion for here and here. So if I started, let's say, just for argument's sake, I start with one cell, you know that one cell is going to divide and become two cells. Those two cells are going to divide, and they're going to divide to become 4 cells and 4 cells to 8 cells. So let's say I have 4 cells that become plasma cells and 4 cells that are memory cells. I started off with 1 cell here, but at this point here, I've now got 4 cells, and those 4 cells can go through that clonal expansion, and therefore 4 each time we're going, that 4 is that 1. So we actually end up with 16 afterwards. Does that explain? What specifically do we need to know about memory cells? Like, what do they actually do? Yeah, so memory cells are needed to basically be the original replacement of the original one that started out, but lots of them, so that when they go through that clonal expansion, it happens at a faster rate. So therefore, you can make more antibodies at a faster rate because you can go through that expansion. Mitotic division to make more Well, oh, that was me saying, so if I kept going, so obviously each one becomes two. I said that I have four that become plasma cells and four that become memory cells. I was just making numbers up. So therefore, what I'm saying is I started over here with one cell. Well, over here, I've now got four cells. Those four cells are going through clonal expansion. That's going to mean there's going to be lots more of them at a much faster rate. So clonal selection would happen, but they didn't want it in this question. I did that. I did the homeobox gene session, either last one or the one before. So if you want to check my live, I want to say it was 2 days before. Check that live out. I did a lot on the homeobox and lac operon. Tony, I'm going to go through the nitrogen cycle tonight. I've got it ready to go. I'm just about to go through that one. Wait, I still don't get why E has the highest number of memory cells. Because you've expanded. So if I had four and I expand, we know that four doubles. So four is my starting. I times that by two. Let's say it doubles 16 times, 2 to the power of 16. What would you actually get? So, oh, it's because I've got my phone here. Hold on. Let me just look at a calculator. Hold on 1 second. So if I've got 4 times 2 to the power of, where are you? That's not what I meant to do. Okay. 4 times 2 to the power of, oh, can I use this one? There we go, 16 equals 2,602 1,144. Yeah, does that make sense? So the key thing is it's where you're starting off at. And so it's to do with those T helper, the cells starting. If you've got 4 memory cells, then going to double each time, you're going to end up with more of it. Okay, I just want to go through one more bit here before I move on to the nitrogen cycle. And I also want to do percentage efficiency. This question still, again, this was 2023 AS paper, and I found it so fascinating the way they asked the question. So this question was looking at you being told about this enzyme will remove the methyl group from the cytosine base, and they're removing it only when it's next to a guanine base. So therefore, they're checking your knowledge from module 2. Do you know what a cytosine base looks like? So do you know your pyrimidines? And do you know your purines? Do you know that your pyrimidine is your cytosine? And therefore you know your guanine is a purine. So therefore you should know that this one is a double ring. And so straight away I can see that I've got a double ring here, but I've only got 2 hydrogen bonds. So therefore this one has to be adenine. I've got a purine here. Opposite way around. I've got my purine above, which is the double adenine. I've got my pyrimidine down here, but I've got 3. So this is cytosine, but it's cytosine next to adenine. I've got another pyrimidine, but it's only got 2 hydrogen bonds, so that's methymine. I'm not answering this question. I'm just saying the bases. A, C, T. I've got 3 and I've got the double, so therefore that's guanine. I've got 3 and I've got a single ring. So, oh my word, I'm writing the wrong things down. That is a cytosine, and I've got 2 and I've got a single, so that's the thymine. So therefore, my cytosine, which is next to a guanine, is this one here. So this is the base that they wanted you to label in this question. So this was a really fascinating way in which what they were actually assessing was module 2 knowledge about the structure of DNA, and what they were also checking was, do you understand how that applies by the hydrogen bonds? So I thought that was a really interesting question, and I really liked it, so I wanted to show it there. Okay, so looking here then, remember, they haven't asked very much on energy efficiency in a long time. So excuse me, you should know that you have your trophic levels, you have your primary producer that's going to be doing photosynthesis, you've got your primary consumer that's going to consume the organic compounds that are in the producer, and they're going to then build them up to their own organic compounds. You've got your secondary consumer, you've got your tertiary consumer. Yes, this is OCR for those of you who are just joining. So the key thing to note is whenever we talk about the energy transformation, energy moving through a food chain, we're talking about the fact we have to remember that these producers, the primary consumers, the secondary consumer, they are all made of organisms that are made up of cells that are going to be doing respiration, because we need that ATP, the ATP for the metabolic pathways and reactions that need to occur. So there is a high metabolic demand in our animals and also in our plants. So therefore we need our transport systems. So important to remember, respiration is always happening. Therefore, there is energy being lost to the environment. Excuse me. Also, not everything that's in the organism, for example, in the producer, is actually going to be fed upon. So through death of the organism, through leaf loss, through excretion, you are going to have material that was ingested, which is not being utilized in that primary consumer. So therefore, what we want to look at is our GPP. That's literally what I'm doing now. The GPP is the gross primary production. I'm going to show you in an example, and I'm going to show you it with some numbers. Now, the exam question that I'm going to give you is not from OCR, just to be clear. The only question they've ever given was in, I want to say, a specimen paper, but it was numbers in a table. I'm going to show it in a diagram that came from a different exam board, but it is very clear that it's there. So my gross primary production is the total energy, that energy, the light energy that can be fixed by photosynthesis, and it's very small. So if in my example, I have 1 million joules of sunlight, only 10,000 joules are actually going to be stored in the primary producer. So the gross primary production is the total energy that can be fixed by photosynthesis. Now, the net primary production is the total energy that's fixed as biomass, mass without water, that dry organic matter. So therefore, what we need to understand is if they ask us about the efficiency, we need to know how much biomass is being transferred, how much biomass was potentially going to be taken in, and times that by 100 because we're talking about a percentage efficiency. So how do we work out our dry organic matter? Well, we would dry our specimen in an oven at 80 degrees C, and therefore that's a way in which we can measure the dry mass that is actually within the organism. So we're basically evaporating, thank you, Amna, evaporating the water out of the cells. So we're left with just the dry matter. If they ask you about measurements, if it's inland, it's going to be grams per square meter. If it's within water, because we're talking about volume, it's grams per cubic meter. So do make sure you note, even though it's not showing it in the right way, that it should be up here. This is where when I transfer things across, it doesn't quite go in the same way. So let's look at an example. So here's an example of a question that they could ask you. So the diagram shows the transfer of energy through a grassland food chain, and all the values you've been told have been given in kilojoules per meter squared per year. So if they're not giving you the units, they may ask you to actually state what that is. They've done that in the past, so they expect you to know joules or kilojoules per meter per year. Yes, I do think evolution is coming up. Oh, wow. Well, this is why I built my channel, so that anyone who's studying OCR can go watch the whole course that's there. Anyway, back to this. They want in this question us to calculate the percentage efficiency of the energy transfer from the producer to the primary consumer. So we know in the question, they've told us that we have got 1.4 times 10 to the 5 in the producer. They've told us that 4.4 times 10 to the 4 is being lost. That energy's gone. They've also told us that 8.4 times 10 to the 4 goes to decomposers. So therefore, if I'm looking at the percentage efficiency that's going to be moving, I need to do some calculations. So the first thing I need to do is I need to know that I had 1.4 times 10 to the 5, and I want to minus 4.4 times 10 to the 4 plus 8.4 times 10 to the 4. So I'm going to minus those two together. So therefore, when I have that, I can work it out as it's 128,000 that is actually going to be lost. So therefore, what you need to do is understand that if I've got 140,000 and I'm minusing 128,000, I therefore have 12,000. That's the difference. So my net primary productivity, oh, my word cannot speak. My GPP is my total, and that equals my NPP minus my respiration and the feces that will be decomposed. So anything that's been lost that's going to be decomposed is not available to the next level up. So therefore, I need to divide this by my total, which is 140,000. And remember, it's a percentage efficiency. So I'm going to times that by 100. And that should give me an answer, which is 8.57142, 8.57142. And so therefore, I would round that to 8.6%. Do you have to know the equation of GPP? The GPP, they tell you, what they're expecting you to understand is that the GPP is going to be your MPP minus your respiration, but they'll give you those bits of information, but you're looking at what's available to the next level, which is really important. Okay, so can I explain crossing over? Guys, if you go and check out my videos, I'm not going to do crossing over right now because I want to go on to the nitrogen cycle and I want to show you how a question could come up linking the nitrogen cycle and ecosystems and the different environments. What's respiratory loss value, Tony? Going back here, respiratory loss value would be this 4.4 times 10 to the 4. I'd make that assumption because it's been energy lost and therefore these are all going to be doing respiration. So I would assume that this is all to do with respiration. So respiratory loss is the 4.4 times 10 to the 4. But you've got to remember that some of the materials not available to the primary consumer because, for example, leaf loss is going to, the leaves are going to drop down to the ground. Well, the decomposers then have the available sources there. So they will digest them down and therefore they will get them. Hence why it goes to the decomposers. I'm going to have to move. Hold on one second. Let me move what I'm using. I've lost my tripod. So I'm now using a cup to hold you guys up for an image. Right. Okay, let's look at the nitrogen cycle. Where is the, hold on, 12,000. Just to go back to this, it was these two. 140,000 minus 128,000 equals 12,000. Jake, Amna's not been a know-it-all. Amna has been in my sessions every single live that I've done, and she has worked her absolute socks off to show, and she's trying to be helpful. So she's actually not being a know-it-all, she's helping you, and she's also consolidating her own knowledge. So I actually appreciate Amna being here. And if it wasn't for Amna emailing me with some detail about what was in the exam, I wouldn't know how to help you guys. So actually, don't say anything to Amna. gonna have her back. She is the goat. Absolutely. Obviously behind me, right? Okay. Right, coming back. So with regards to this one, sorry, yeah, I tried to do it on TikTok, but it didn't really work. So guys, if you're just joining me on TikTok, jump over to my YouTube channel, because that's where I'm showing the questions. It's on right now, live right this second. Um, right, I want to go through this question. So this question was actually a question which was in a GCSE, um, GCSE, international GCSE paper, and I've modified it to make it an A-level paper. And look, it's quite funny actually. You can see, look, I'm going all red because I'm not well, and also because I'm like really passionate. I know how close you guys are getting to that end point. Vasodilation in its prime example. How funny is that? I'm going really red. And it's a thing that a lot of people would be like really embarrassed about things like this. And actually, it's one of those things that it's a natural biological process that happens because I'm getting excited because my adrenaline is rushing. It is therefore causing my blood vessels to dilate, my arterioles. So there's the biology behind it. And you'd be surprised how much of that will come into play in your exam when you're in that exam and your adrenaline's pumping and you're stressed and you're like really going for it. What's going to happen is you're going to end up with that vasodilation, and it could be linked to inflammation. You're absolutely right. Thank you, H. The key thing is because I've actually moved on to being online now, it's why I do these sessions for you, so I can help as many as I can. I'll probably go on for about another half hour, and then I need to give my voice a rest for my students I see tomorrow. So yeah. Oh, you're very welcome. You're very welcome. See, you've all got these bits of information inside you. When it comes to the exam, you're going to see that you can get it out. It's just about giving yourself the time to go, like, I do know this. Be a show off, be a know-it-all. Because actually, what you're doing is you're showing off to the examiner, you're showing off to the person that you know your stuff. That's the important part. Right. Okay, let's look at this one then. What have we got then? We've got some information about a nitrogen gas that's in the atmosphere, 80% in the Earth's atmosphere. We're being told about recycling of nitrogen. We're being told about these nitrogen containing compounds. And we've been told about this small farm in Ghana. So they basically linked it to a novel idea. You've got a small farm in Ghana. How is that got anything to do with this recycling of the nutrients? So we know within our ecosystem area that there are a whole load of nutrients that need to be recycled. So they're either going to go down the carbon cycle and carbon dioxide side of things, or they'll go down the nitrogen cycle, or they'll bring the two of them together. So it's important that you do note that they go together. Now, Jake, is an interesting one. You're saying about plant physiology still on the spec. Yes, it is. But the way they ask the question will be about, and it's an interesting one because I have a question to show you. It could be asking you about the cell differentiation, which is module 2, and you should know about how your plant cells, your hair cells are differentiated to be able to do their job. So if they're asking about nitrate uptake, they're actually talking about cells differentiating in the root so that they can take that nitrate ions through across the phospholipid bilayer. Because remember, to get into a cell, you have to cross a biological membrane, the plasma membrane. So they could link in your biological membranes topic as well while looking at the nitrogen cycle. How do nitrogen and carbon link together? Well, if we're talking about amino acids, we need to understand that the amino acids come from the fact that we've got an amine group and we've got a carbon skeleton. Where did that carbon skeleton come from? Well, remember our triose phosphate that was produced during the Calvin cycle of photosynthesis, that can then link together. So it's about the way in which they want to put it. Yes, they do actually, Lisa. You're absolutely right. There is a question that says, how are they similar and how are they different? You're absolutely right. on that one. Right, let's go to this question then. So we're being told that we've got nitrogen in the atmosphere. We're being told that we've got ammonium ions now in a bacteria, which is in a root nodule. So therefore, if the nitrogen has been converted into ammonium ions, we know that this is nitrogen fixing. So we've got nitrogen fixation that's occurred because the nitrogen has been fixed to make ammonium ions. And in the exam, they will allow ammonia and ammonium ions. However, they will not accept you writing the wrong information with regards to your chemical formula. So make sure you know that it's NH3 for ammonia, NH4 plus for ammonium ions. Just be aware of that. Then what they're telling us is that ammonium is going to happen with regards to, so nitrogen fixation, what we're using is we're using bacteria. Bacteria are able to do this aerobically and anaerobically. It's more, if they're looking at the anaerobic side of things, you're absolutely right, Nux, we're looking more at denitrifying, because if there's not enough oxygen there to be the last electron acceptor, then we would denitrify. But remember, bacteria are able to do aerobic and anaerobic respiration. So it's not that it's not possible, but it depends on which way they want to go with the question. Yeah, so it can be both. It depends where they want to go. So it's mainly anaerobic because that's why you have the symbiotic relationship with the bacterial cells and the plant, because the bacterial cells get not only nutrients, but they also get a supply of the oxygen. Yes, but you have to write it correctly. So if you're doing the nitrite, you have to do it with a minus. So looking at how this is going, we've got our amino acids. We've got amino acids in the plant, the legumes. We've then got proteins in the legumes, and then we have the amino acids in the intestine. So Q is digestion. Those proteins need to be hydrolyzed down. They're hydrolyzed in the intestines of the goat so that we now have the amino acids, the smaller monomers, which can then be absorbed into the bloodstream. So then what you have is amino acids to create the urea. Well, we know deamination occurs, because remember, this could come up in paper 3 as well. So it's important you also know that it could come up in paper 3. Yeah, so that's the reason why if it's taken away the other option, which is the denitrifying one, that's what they'll be looking for. S is protein synthesis. This. And then we're looking at the fact that we've got urea and the protein in feces. So this could be decomposition, or it could be ammonification. I'm answering my YouTube stream. I'm sorry, guys. I was on here with it displaying, but it crashed on me, so I had to close this one down. Okay, so T is decomposition or ammonification, and then we've got U, which is nitrification, and V, which is denitrification. So they could ask you to do that. So just as a quick reminder, nitrogen in the atmosphere and nitrogen fixation, your two types that you need to know. I see some people writing it. Rhizobium in the root nodules, azotobacter in the soil. That's then absorbed and used to make amino acids. Amino acids are then made into proteins by doing a condensation reaction. Consumers need to break them down. And then if they're breaking them down, they then need to build them up. So you've got digestion and protein synthesis. Ammonification is basically the process of what decomposers do. When we are converting the dead material into ammonium ions, That is a modification. How do you find the critical value? It depends on your degrees of freedom and the test that you're using. To answer that question. You then get ammonium ions are going to be nitrified oxidation reaction to give you your nitrite. And that is, as someone rightly said, nitrous ammonius. Again, another nitrification, nitrite, nitrate. That's from nitrobacter. So that's how I remember it. Nitrate as an A, nitrobacter as an A. And then you have got that being absorbed into the roots. Once it's absorbed into the roots, it can then be converted into amino acids and then built to proteins. And then you've got your denitrifying on that front. So then they could potentially so Jamie, to answer your question, what you have to understand is nitrogen has, you're absolutely right, it's got 3 bonds. It's got 3 bonds, but it's also a molecule which is pretty stable because of those 3 bonds. The oxygen only has 2 bonds. It's a much smaller molecule, and therefore it's able to move through across the alveoli into the erythrocytes. But remember, the only way it can move across is if there is a concentration gradient. So the diffusion is that concentration gradient of moving across, whereas the nitrogen can't move across. Nitrogen gas just basically stays in and comes back out again. So it's not able to cross the barrier. Karina, if you guys are just joining me, what I would say is go and check my video. If you want something specific, I have done a session on my live yesterday or the day before on Hardy Weinberg, but I've also got videos on Hardy Weinberg, so do check them out. No, Shane, no, you do not need to know about RNK strategists. That is not new at all. That is really, really old. And videos that say they're OCR that are still talking about RNK strategists, they're way past their needing to be revamped. Don't worry, RNK strategists are not involved in this at all. Patterns of inheritance, yes, I feel is coming up. Manipulating genomes, yes, is either gene therapy or genetic engineering that's coming up. So naming those ones could be asked. And then this question is the one that I really love and I want to talk about. Liver neuronal is not paper 2. Liver neuronal is paper 1 or paper 3. Hormonal neuronal will come up again, but technically neuronal kind of came up with a cardiac cycle question. Oh, sometimes they do give you a bit more, but it's not part of this. It's really not. I had a student who sat and watched a 2-hour video 2 years ago because obviously this was before I started my channel, when I first started teaching OCR again. And they watched a 2-hour video, came in and started talking to me about RNK strategies. And I was like, what are you doing? It's not part of your spec. They're like, but it's OCR. They say they're OCR. I was like, yeah, but it's not relevant. It was videoed like years ago. Why are you doing that? So there was a lot of applied. Overall, the application can be anything. And this is where you have to look at your specification, guys. They actually tell you in the specification where they could ask the questions and where they'll go. So give me 1 second till I find it. In the actual specification, oh, I'm gonna have to stop soon. My throat's getting really sore. Oh, I just went past it. Yeah. So this tells you the percentages that they could give you on the different sides of things. And a lot of students don't actually realise that, if I put that up there and then put this here slightly. So what you can see is A1 is about your knowledge and understanding. That's your memorization. That's small. If you're looking at the percentage, 31 to 34% on AO1 on your content. So only 13 to 14% of your paper will be based around AO1. There's no topics that have been taken out. They've just specified what is important. So you don't need to worry about things that have been taken out. What you need to do is you need to be using the current specification and only looking at what it states in there. So they've just made it a lot more understandable for you as students and also for us as teachers as to how much detail we need to go into. Do you need to know about euchromatin and heterochromatin? No, you don't need to know it, but you need to understand it. You need to know they could ask questions about peat bogs, but they're not expecting you to have that as a case study anymore. But if you are trying to cram stuff, please do check out my videos. I really want to cover this question. One second. So one question I want to really show you guys, which again is not from your exam board, but you hopefully have done lots of questions in your exam board you're now done with. But this is where they could take, because they're never going to ask the same stuff. What they're going to ask is other things. So if they give you a graph, and this one is talking about nitrate ion. So this links really nicely to the nitrogen cycle and talking about the absorption in plants through the roots. So they're looking at, do you understand what happens in the roots? And do you know you've got your root hair cells? So do you know how things can be absorbed into the roots? Do you know that the nitrate ions are made in the nitrogen cycle? So it links to nitrogen cycle. And then they've said, right, well, what we've done is we've got these nitrate solutions of different concentrations. And what we're then going to do is ask you to use this information to pull out from it and say what you see. Now, they're only interested in these two, and they're only interested in the hours of 2 hours to 5 hours. So make sure that you know if you see a straight line, this is a constant rate of uptake because we've got that straight line. So do make sure that you can talk about that in your answer. And also make sure that if they then go on, which is what I want to show you, if they then go on to show you a table of results, I see so many people say to me they don't quite understand how to pull information from a table of results like this. So last year they did it all on, it was the donor leaves being taken away, and it was about the mesh bag, or it was about the photosynthesis practical. Whereas this one's talking about nitrate uptake in roots, and they've given them different conditions. So if we start off, the first thing I would always say is start with your control. That is your place that you're going to compare to. So let's have a look at what they've done. We're going to start with number 1. And what have they told us? Right, well, the temperature was kept the same. The difference was there was no aeration. There was aeration. There was a much lower uptake of nitrate when there was no aeration. And this is because there was nitrogen gas that was bubbled through instead of oxygen. So if there's no oxygen in the solution and there is no aeration, therefore there is no oxygen. What is oxygen needed for? Well, oxygen is needed for aerobic respiration. And I know a lot of you will then go, oh, but they had so much in paper 1 about respiration. They had so much in paper 1 about respiration. but it was really about enzymes. And it was about the intricate parts of respiration. If they're asking about it here, what they're looking for is you to understand, well, aerobic respiration is about making ATP. So if I've got no oxygen, I've got no aerobic respiration, I've got no ATP. So if I've got no ATP and the uptake of my nitrate is really low, what could I therefore assume? So at the moment, I'm going to make an assumption. If there's no ATP, there's no active transport. So if there's no active transport, therefore, I have not got any uptake, and that's because there's no ATP. But I need to prove this. So that could be one way in which I could draw a conclusion. So this would be where module 1 comes in. Because remember, module 1, 2, 4, and 6 is in paper 2. Module 1 is about how you are able to draw conclusions from tables of results. So in this case, what we're saying is there is no ATP because there was no aeration. I can make the assumption there's no respiration occurring, no ATP, no active transport. But let's just see what else happens. Right. But if we look at condition 2, we can see that the temperature has dropped down. We have a low temperature. We do aerate this time, so there is ATP that's being produced. But because there's a low temperature, that low temperature is therefore saying that there is less kinetic energy. If there's less kinetic energy, I'm going to expect there to be less metabolism, metabolic reactions. So therefore, I would actually say that there's less ATP. So it's important that you are able to apply your knowledge, but this is actually assessing your module 1. Yes, Karina, I do, but I also have a few lives on the zero dilution side of things, but there's definitely lies out there somewhere that's on zero dilution. But if you go to biotechnology and you go to culturing microorganisms, I think is in that video. Looking at this next one, protein synthesis has been inhibited. If I've got a temperature of 30 degrees C, I've got aeration, but protein synthesis is inhibited. I can see that there's a reduction in my nitrate uptake compared to my control. So therefore, proteins are required. Well, proteins are required for active transport. So therefore, that makes sense. If there's less proteins being made because they're being inhibited, therefore, there is going to be less movement. We're inhibiting the proteins that are going to do that. And then the last one, well, this one, it's dropped down, but it's only dropped down by 0.9, and antibacterial are going to kill bacterial cells. So therefore, if we've got something that's going to kill the bacterial cells, but it's not really having an effect, bacteria are not involved in the movement of the substances across. I don't think succession, Tom. So Tom, you just asked about ecosystems, mainly succession. Succession was in last year's paper. I don't feel like it will be that high, but ecosystems will come through potentially on nitrogen cycle, carbon cycle, if they're going to do that. But I'm just guessing. I could get it wrong. No, Harry, the 93 to 3 to 1 ratio is you assume that there is no linked genes. Crossing over only occurs when there's linked genes. If you've got crossing over occurring, you would not get a 9 to 3 to 3 to 1 ratio because the genes are linked, so they wouldn't give you that nice independent assortment, and therefore you would have probably high numbers and low numbers in your threes. So your threes would go down to ones and higher elsewhere. No, you don't need to know the phosphorus cycle. Do you think mitosis will come up this year for paper 2 again? Yes, because mitosis is needed if we're looking at the eukaryotic cells and it's part of cell 2. I think they'll probably get you to draw a diagram maybe on that one. I don't know. A 9:3:4 ratio would be if it's epistasis. So yeah, this one's linked to nitrogen cycle, but it's also linked to cell differentiation, so module 2. Because we're talking about roots and root hair cells. We're also looking at biological membranes because we're looking at the movement of substances across a membrane. And so that's why I wanted to show you this, because a lot of people say, oh, biological membranes hasn't come up. This is the way in which they would ask it, but it's linked and looks at a different way. Are you supposed to remember the ratios? You're supposed to know how to work the ratios out, but you should really know your 9:3:3:1 ratio. This is OCR, OCRA. This exam board that this question came from was CIE A level. Okay, I'll finish on my list of what do I think could potentially come up. My little book I've got here that hasn't come up. Where is it gone? Right, if I were to make a prediction based back when I did it, looking at all the papers, my predictions for paper 2 still to come up could come up in paper 3 if not, but paper 2, batch and continuous fermentation. I did say immobilized enzymes, but I'm umming and ahhing over that one at the moment. Lycopron transcription factors, artificial cloning with the animal side of things potentially. They didn't do a dissection yet, so I feel like a dissection might come in. Natural selection and speciation, plant disease and defenses, nitrogen cycle, I've already mentioned, DNA replication, DNA sequencing but linked to genetic engineering, gene regulation, which I've already mentioned, differentiation, percentage energy efficiency, and evolution in fisheries. Is what I would say is my prediction of what may come up based on my previous analysis of the papers. But I don't know. They asked a large question on immunity last year. I feel like they're going to go down the plant side of things, potentially with the communicable diseases side of things. I do feel like phylogeny and evolution potentially coming up. Gene flow is about the flow of the alleles. Genetic drift is talking about a chance occurrence, some natural things. So it's not natural selection. It's about changes to the allele frequency that is not due to it being advantageous. So genetic drift is like the bottleneck effect or the founder effect is those two. What do you think about equivalent to 30 grade ranges? Why? I think it's probably about on par with last year's paper. Cell mediated is your T cells. Humoral is to do with your B cells. Yeah, I feel like the evolution, they could look at DNA that has come from an extinct organism and they found the DNA. They've then analyzed that DNA. They're looking at the DNA of current species in a particular area. They show you a graph of a migration and they want you to then link that all together, potentially. Paper 2 is module 4 and 6, but 1 and 2 will come through. If you don't understand module 2, then none of the rest of it makes sense. Everything to do with module 4 and module 6 is all about module 2, but in context. So if we're talking about for example, antibodies. What are antibodies? Antibodies are proteins, proteins that are made through protein synthesis, transcription, translation. What's module 6? Module 6 is all about nucleic acids and nucleotides and how that applies and how we can manipulate it and how we can use it. Biodiversity, module 4, link into module 6. So what is module 6? Ecosystems, nutrient recycles, that's biological molecules. So all of it is interlinked. So if you are really confident with your module 2, module 4, module 6, module 3, module 5, all of that is just putting that into play. I drilled and drilled and drilled, always going back to module 2, always going back to module 2. And I had a student who, when I first started teaching them at the beginning of year 13, and they actually hated, they were like, who is this person who's teaching me? Because of the way I teach and I ask questions, and I'm constantly referring back to all those different modules. At the start, they hated the way I taught. By the end, they were literally like, wow, Christine, you've like opened my eyes to how the world is and how biology is interlinked. And what that therefore meant was when they went into that exam, they were able to pull their grades up because they could find those linking points, which they never put in before. And they were an intelligent student. They just never put the basics in. And so it's important to know that go back to basics and build up from them. Micropropagation, if you haven't seen it already, I have a video on cloning in plants specifically. Management of ecosystems is basically what you're going to do. You're going to stop grazing, because grazing, if you allow too much grazing, that is not only going to deflect succession, but it therefore is going to affect the species and their food sources. So you've got a real knock-on effect of these food webs, food chains. So management of it is about how often you can do it, because you want to allow it to be able to continue to grow, because plants need to be rooted so that they anchor in. Because if you don't, what are we dealing with right now? A lot of landslides. By deforestation, we're removing plants, removing trees, we're removing things that could have a knock-on effect to the rest of the ecosystem. You need to know the examples, you need to know what they stand for, but if they're going to ask you a question, they will link it to an example. I feel like they did a lot on that last year, so I don't feel like it'll come up that much, but it will be there somewhere. Amna, awesome. You're gonna nail this. Yes, I have. Oh, no, you're asking Karina. I'm like, yes, I've seen those papers. Which practicals you'd have to look through, but there's so many that they could ask. It really just depends. Right, guys, I am going to finish. My phone's about to die, and my throat needs a bit of a rest before I do my sessions tomorrow. I will try and jump back on here tomorrow night when I can. I need to put my girls down. It's my night to put them down. So I will try and jump back on here to help you. But if I don't manage to, good luck, okay? I should be able to come on here tomorrow night. But just in case I don't, for those of you who might not be able to come here or might want to just get your head in your own headspace, good luck for that exam. Just take your time, read the question. That's what I would say to you. That's the important part. Oh, Chidima, hi. Sorry, I just saw you on here. Right. I am now going to stop. Good luck.